Yes. All right. And after each class, I'm gonna send the link to you, so in case you want to watch again. Okay. All right. Any other questions? No. Okay. Perfect. Okay. All right. All right. So this slide only shows a example, the marker map in Sandy Morgan, in the barley genome. So obviously in barley, so there are a total of 7 chromosomes, and as you can see, there's a lot of markers genes along each chromosome, and this doesn't show the real map, physical map. It shows the genetic map measured in the Sandy Morgan. So yeah, so this is so in this class, we're gonna talk about how we're gonna do the genetic analysis for the genetic map, not the real physical map. Because if you are interested in the sequencing and the real map stuff, so I think you can find some other classes in the department, and you can learn the physical, how to get the physical map, and the related study from those classes. All right, so for our class, we focus on the study genetic analysis of the genetic map. All right. As I mentioned, we have two measurements, right, for the genetic map. One is using the number. Oops. One is using the numbers of the crossovers. For example, so we're gonna talk about this more later. So if we want to do this, we have to use some experiments. For example, the test cross. So I believe most of you have already learned about this in the basic genetic classes. So if you do a test cross, you're gonna generate a lot of progeny. And in each progeny, so each progeny represents a single meiosis event. So if you look into the cell, each progeny, or if you just look at the phenotypes of each progeny, you're gonna see something like this. So for example, the first one is one progeny. So if you look at the genotype, let's see. So the test cross, we have so the test cross involves 2 loci, A and B, and they are linked, of course. So they are located on the same chromosome, and we want to see how far they are removed from each other. So then, so for 2 loci, we have the double heterozygous loci, or we have a term, the dihybrid. And so when we do the test cross, so if there's no crossing over occurring between these 2 loci, you're gonna see the same, right? And we call this parental type, so because there's no crossover in between. However, if there's a crossing over, a single crossover between A and B, so what you can see is in this progeny, in this meiosis, you're gonna see different genotype. You're gonna see probably different phenotype as well. And this phenotype or genotype are different from the parental type, the parents, the genotype of the parents. So we have a name for them. We call them recombinants, right? So they are different from the parental. And okay, so if there's only one crossing over, you're gonna see the non-parental type, right? The recombinants. But if there are 2 crossing over, although this is very rare, but it could happen. So if there are 2 crossing over occurring between A and B, so what happened here? So you're gonna see the parental type again. So which means you don't see any recombinants, right? All right. If So we're talking about the first measurement for the genetic map. So we use the number of crossing over in between A and B to calculate the genetic map distance between these two loci. So just imagine, so I just listed three progeny or three meiosis. If we have like many thousands of this kind of progeny, if we look into each of these meiosis to see how many crossing over in between A and B in each meiosis, we can do the calculation, right? So if we do the average, we can calculate the average crossing over between this A and B. So that's the first measure of the genetic map distance between A and B. Is that clear? Okay. All right. So this is something related to the biology. So if A and B are tightly linked, if they are on the same chromosome and they are very close to each other, then the probability of having two or more crossing over in between is very small. So for example, this one or even more crossing over occurring between them, it's very small. So the most frequent occasion you're gonna see include these two. So no recombination, or there's a one crossing over. Okay, so this is probably this is very familiar with you. So I taught genetics, very basic genetic class for the undergraduate student. So this is the picture, the classical picture I often use to teach them how a test cross is performed. So we start from two parental lines, pure lines. So for the parent one, you're gonna see we're talking about two loci A and B on the same chromosome, right? So the one parent is homozygous in two loci, and another parent is also homozygous in two loci, but this parent are carrying the recessive alleles, for example. So this parent is carrying the dominant alleles. All right. So now, if we do the cross between these two pure line parental lines, we're gonna get a dihybrid F1 individual, and then we want to use this to cross with a tester. So the tester is similar with this parent because at two loci in this tester, the two loci are carrying are both carrying the recessive alleles. The reason why is that because
clarification.
Yes, go ahead.
Just
right.
Making absolutely sure. So the double recessive for the tester and the original pure parent line, these are the same alleles, correct? Because I noticed
Yes.
they were colored.
Of course, yes, yeah, that's right.
I wanted to make sure.
Yeah.
Okay.
Yeah, because yeah. So but yeah, this is a good question. So in reality, in the literature, so it may not be the identical. But for this example, taken from the undergraduate student class textbook, so it's a very simple one. So we don't want to make a very complicated situation for them to understand. So here, the same allele, yeah, the same allele. But you see the color is a little bit different because we want to differentiate this tester from the original parental line here. But the allele are the same. The reason why is that because we want to use this tester which cannot hide anything from this F1 parent, right? Because we want to just show what is happening in this F1 individual. All right, so when we do the test cross, we're gonna produce lots of progeny, because we need a big data to do the statistics. But even we have a lot of many thousands of the progeny, this progeny will fall into four categories. All the progeny will fall into four categories. All right, if you look very closely to these four types, so these two types are called the parental type. Why? Because they are showing the parental phenotypes, right? And you're gonna also see two other types. They are showing different combination of the two traits. So why is that? Because of the crossing over between these two homologous chromosomes, right? So there's a crossing over occurring between these two loci in this in the F1 individual. Then the consequence is you're gonna see some progeny showing the phenotype which are different from their parents. All right. Okay, so this is a very classical experiment showing the test cross. We're going to use this as well as a tool to help us to calculate the recombination frequency. Now we are talking about the second measure. In the previous slides here, we're talking about using the number, the average number of the crossing over to measure the genetic distance between A and B, right? And here, so we try to calculate another thing, the recombination frequency, which can also reflect the genetic distance between A and B. All right, so this is another example I often use in my fundamental genetics class. So suppose, so we want to check the genetic distance between two loci. One loci is represented by letter B. So there are two alleles. The big B, I think, the big B is the dominant allele, controlling the purple color, and the small b is the recessive one. It will render the red color. I believe this is an example for the eggplants or something. Yeah. And the L, the capital L is the shape, is the length, is showing the long in shape, the eggplant. And the little L is the round shape. So these two genes are controlling two. So this is two traits that are controlled by two individual genes. And because these two genes are linked, they are on the same chromosome. So then we can do this kind of test cross experiment to help us to measure the genetic distance between these two loci, two genes. So following the diagram I showed in the previous slides, we can do this kind of experiment, right? So we can generate a dihybrid parent, the double heterozygous here, and we test cross this parent using this tester, so the double recessive, and this is what we can get from the progeny. And obviously the same thing, right? So we can get 4 categories here. So 2 categories labeled with P means they are showing these progenies, these 2 groups of progenies are showing the same phenotype with the parent, but the 2 categories in the middle, they are not. They represent the recombinants. They represent the crossing over, all right? Now, so based on this data, so we're gonna generate a lot of progeny here. Just for simplicity, we use a very simple number here. We assume we just get 100 progeny in total, but in reality, this number is not enough. So we need to generate more, way more than this to get a robust estimation from the statistical analysis. All right. Now we can use this very simple equation formula to help us calculate the genetic distance between these 2 loci. So it's very easy to understand, right? So the numerator is the number of the recombinants. So here, there are 10 in total. So you put 10 here, and the denominator is the total number of the progeny. So we're gonna see what's the percentage of the progeny showing different phenotypes from their parents. All right. And so this is the calculation, very simple calculation we can use to calculate the genetic distance between B and L. So there's a 10 map units here. Sally Morgan. Any questions? Ryan, you have questions? No? Okay. Very good. All right. So now, so I hope so each time I try to slow down because I try to make sure there's a logical linkage between the first slide, the previous slides, and the current slides. So in the previous two slides, so this one, I showed you how we can use the average number of crossing over to measure the distance between A and B, right? So here we try to use another thing, the recombination frequency, another measure to measure the distance between A and B. Okay, now since we have two measures, what's the relationship between them? So this slide shows the relationship between them. As you can see, so this graph, beautiful graph, the x-axis represents the genetic distance measured in number of crossing over. So this is the number. So for example, 0 crossover, 1 crossover, 2 crossover, and so on. And this measure is a additive map distance. Intuitively, you can see that is correct, right? So the measure in number of crossing over is a active, sorry, not active, additive, is an additive map distance. Now, if you look at the y-axis, so this is another measure. So if we use the recombination frequency to measure the genetic distance between any two loci, so this map is no more the additive map. So we're going to talk about this because one map distance is additive, the other one is not. So you're going to see a nonlinear relationship between these two, right? So here we try to find the relationship between these two maps. So this is called the map function. All right. This is the major point for this chapter. Any questions? All right. So we have two famous map functions try to explain the relationship between the additive crossing over based map and the recombination frequency based map. So the name is here. The names are here. So one is the Haldane map function, and the other one is Kosapi map function. All right. So if you look at these two map functions, so showing the relationship between the additive map distance and the non-additive map distance. At the beginning, when we talk about the genetic distance between two loci, if the distance between two loci is very small or very large, so these two functions are agree with each other quite well, right? So they are very close to each other. The only distance between these two map functions is in here. So which means, yeah. So in two extreme case, when the map distance is very small or very large, these two functions will give you the same results. But if the distance between two loci we're talking about is not too small and not too big, then you're going to see difference between these two functions, map functions. So we're going to look into these two functions and to see why they are different. They're both very useful, but yeah, they're different. So we're going to see the details. All right. Okay, so let's look at this example. Suppose we are interested in finding out the genetic relationship or the genetic map distances between these three loci, which are located on the same chromosome. Suppose this is the real situation. So A, B, and C are linked like this. And as I just mentioned, if we use this map, so this X represents the number of crossing over. So this is one way we can use to measure the genetic map distance, right, between A and B, or B and C, or A and C. Since this measure is an additive measure, so we can use this formula. So the average crossing over between A and C, this is one way to measure the genetic distance, is equal to the average number of crossing over occurring between A and B plus the average number of crossing over occur between B and C, right? So this is how we explain the additive formula here. Any questions for this? Any problems? However, when we use another measure, so we can use R, which is the recombination frequency between A and B, between B and C, between A and C. So the additive relationship between these three are that doesn't hold. So this is not true. So that's why in the previous slides, we see when we try to plot those two measures in a single plot, you do not see a single line, right? You do not see a linear relationship because one measure is additive, the other one is non-additive. Okay. Now we're gonna use some like probability theorem to help us to do some simple calculations here. All right, so if we want to calculate, so find out the genetic distance between A and B and C, right? So we need to do what? We need to do the test cross. And we need to use this test cross to help us to produce a lot of progeny. And then from this pool of the progeny, we can check either the genotype, if genotypes are easy to observe, to detect, or we can just design experiment to let the progeny show different phenotypes. The phenotypes are linked to the genotype, so we can use this data to help us to find the parental type and the recombinants, right? And we use this to do the map calculation, distance calculation. All right, suppose, okay, so we have, so this is the chromosome and this is the real situation, how the A and B and C are arranged along this chromosome. So there are a couple of situations, possible situations here. So the crossing over could occur between A and B, right? Or it could occur between B and C, or could both. So those are the possible situations. So if we want to calculate the recombination frequency between A and C, so if we produce a lot of progeny from the test cross, we're gonna look into those progeny to find the recombinants, the non-parental type for A and C, right? So does this situation, so is this situation counted towards this calculation? Okay, so let me say that again. So, okay, so if we want to do the test cross and we want to calculate the recombination frequency between A and C, we can only use the recombinants progeny in terms of A and C, right? So if we have two crossing over between A and, oh, sorry. Geez, I used the wrong diagram, sorry. Here, so yeah, sorry about this. Please look at the here. So the B should be capital B here is the capital C, should be capital C. And the small b should be small c here. Okay, sorry about this. I used the wrong, I need to change this one. Sorry about this. All right, so if there are two crossover occurring between A and C locus, A and C, so the progeny will not be a recombinant, right? If we are talking about the loci A and C, is that correct? All right, so when we look into the progeny from the test cross, we can only use the recombinants in terms of the A and C, is that correct? All right, sorry about this. I used the wrong diagram here. All right, now look at here. So suppose the region 1, let's define this region as 1 and this region as 2. Okay, so in what situation the crossing over in these two regions can cause a recombinance between A and C? So there's one possibility is like, so if you have a single crossing over occurring between A and B, but not in the second region, right? this will produce the recombinance between A and C. So if these two regions, let's see, if the crossing over occurring in region 1 and in region 2 are totally independent to each other, so if you want to calculate the probability for this situation, so this is the probability. The R represented the recombination probability or frequency between A and B, right? So this number, the RAB, will give you the probability that we have a single crossing over in region 1. If these two regions, 1 and 2, are independent, so we can simply multiply these two probabilities, right? So if you look at the first part of this equation, so this represented the crossing over, probability of crossing over in region 1. And this probability showing the probability of not crossing over in region 2. So this, the multiplication of these two numbers showing the first situation. You have a single crossing over between A and B, and this will produce a recombinance in terms of loci A and C. All right. So this is one situation. Another situation is the same, right? So you just calculate this. So okay. So if you have a crossing over in region 2, so you're going to use this probability, and this probability is showing that there's none crossing over in the region 1. So you add up these two probability, and this gives you what? This gives you the probability, the recombination frequency between A and C, right? Is that clear? All right. So if you do a little bit algebra, so you can express this equation like this. All right. So how do we understand this? So clearly, so the measure in R, the recombination frequency, is not additive because the RAC equals to RAB plus RBC minus something, right? So why is that? So how to understand that? Because this is very, so if you think about this, it's not that hard to understand this because this number, the RAB, represents the frequency, the probability overall having a crossing over between A and B, right? This probability including two situations. One situation is this situation. There's only one crossing over occurring between A and B, but none between B and C. But this probability also includes another probability situation. We have one crossing over in AB and another crossing over in BC, right? So we have to subtract that probability from this one. And the same thing, we have to subtract the probability for double crossing over from this probability as well. So that's why you see this equation. Is that clear? Is that clear? Okay.
Could you go over that one more time?
Yes, sure. Yeah, this is very complicated. Let me go over this one more time. All right. So first, so let me ask you. So in the statistics, so when we talk about two independent events, so events A probability equals to PA, right? Probability B equals to for the events B. So if these two events are totally independent. For example, you toss a coin twice. So you can treat these two tosses are independent to each other, right? So then if you want to calculate the joint probability, for example, what's the chance for you to end up with two heads for these two tosses? So you can simply multiply the two independent probability, right? 1/2 multiplied by another 1/2, right? Mm-hmm. That's the chance you're gonna end up with, right? 1/4 is the chance that you're gonna get two straight heads for these two tosses, right? The same thing here. So so this is our assumption. So maybe it is not true for all the cases, but here, if we assume the region 1 here between A and B, and the region 2 are independent to each other, just think about the two tosses here, right? So, okay, now, if the chance of having a crossing over between A and B equals to this, RAB, and the same thing here, if there's a crossing over occurring between B and C equals to RBC, so that's the probability, right? Is that clear? Okay, now, let me know. So what is the probability that you're gonna see double crossing over in these two regions? One crossing over in region 1 and another crossing over in region 2. What's the probability? Yeah, that should be RAB multiplied by RBC. Is that correct?
Yeah.
Is that correct? Because they are independent, right? So if we want to see two crossing over in a situation, so you just multiply the RAB with the RBC, right?
Mm-hmm.
Okay. Now the same. So if you look the first, sorry, let me use this. So if you look at this formula represents what? Represents if you have a single crossing over in AB, but none crossing over in BC.
Yep.
So that's how we calculate this, right? So the first situation. So if the probability of having a crossing over here is this, and what is the probability that you do not see a crossing over here? It's 1 minus this, right? So if these two regions are independent, so you simply multiply these two numbers, and that's the probability for this events.
Okay, no, I get it.
Okay, now, yeah, the same. So for the second event, so this event can also produce a recombinant between A and C, right? The second event. So how do we calculate the probability? The same, because this represented the probability having a crossing over in B, between B and C. And this represent 1 minus RAB represented the non-crossing over occurring between A and B. Both events will produce recombinants between A and C, right? So then we use the rule of addition in the probability, and this is what the real recombination frequency you're going to see between A and C.
Professor, excuse me, why are we not factoring in the possibility of both A and B, both 1 and 2 occurring, like between A and B and C?
Yeah, okay.
Because that also gives like...
Yeah, that's right, exactly. But when you do the test across, so sorry again about this. So if you look at here, if you change the B to C here, right? So we are talking about A and C here in this diagram. Okay, so if you have two crossing over, then you do not see the non-parental type. You do not see the recombinants, right? So that's why we don't count this one in the map distance calculation. So that's why, so when we calculate the recombination frequency between A and C, we only take these two situations into account. Either there's a single crossing over occurring between A and B, none between B and C, or there's a crossing over between B and C, but none between A and B. Yeah. All right. Very good. Now, so this equation just shows this measure in the recombination frequency is not additive, right? Because if it is additive, so we do not have this term, right? But it's not additive. Okay, why is that? Because when we calculate this probability actually includes two parts. So the RAB includes two parts. The first part is this situation. So there's only one crossing over occurring A and B, right? But this probability includes another part, which shows two crossing over. One is in between A and B, another one is in B and C. So that does not contribute to the calculation of the genetic distance between A and C. So that's why we have to subtract that from this equation. And the same thing for the RBC. So that's why we want to subtract twice for the double crossing over. Is that clear? Okay, very good. All right, so as I just mentioned, so this is a very extreme situation. So when we assume these two regions, 1 and 2, are completely independent, this situation is always not true in the reality. Okay, let me give you some very, a situation showing the very real situation. Oh no, so this one is also very extreme. So if we assume the completely independent, we're gonna end up with this. But if these two regions are completely dependent. What does that mean? That means like if you have one crossing over in region 1 between A and B, this crossing over will prevent any crossing over occurring between B and C. This is called the completely dependent or completely interference. All right. So if you have a crossing over already occurring in region 1, so it's impossible for you to see another crossing over between B and C, because the total region between A and C is very short. So it will rule out the possibility that you're going to see multiple crossing over between this region. Does that make sense? So that means completely dependent. So in this case, what happened here? So you're going to see only in this case, this map measure will become additive. Does that make sense? Okay, very good. Okay, now this two examples shows two extremes situations. Now this is something very natural. Okay, so usually, so look at here. Usually what we see in most of the cases, when we calculate the RAC, the recombination frequency between A and C, is calculated using this formula. And if you compare this one with this one, so you're gonna see the difference, right? So you're gonna see the difference is here. There's a coefficient here. All right, so this coefficient we give C as a name, coefficient of the coincidence. All right, and it is related to this term I. So I is the coefficient of the interference. I just mentioned the interference. So the complete interference equals to the completely dependent. So one crossing over will completely prevent another crossing over in the same region. So if I equals to 1, if I equals to 0, sorry, if I equals to 0 means there's no interference at all, right? And then C equals to 1, and this formula will be equivalent to this formula, right? This shows the one extreme case. So if there's no interference, that means region 1 and region 2 are totally independent. Then we're gonna end up with this formula. But if I is not equal to 0, then we're gonna end up with a formula like this. So this C actually controls the interference between, or the probability of having a crossing over in these two regions. So is that clear? Okay. All right. Now, map function. So we're gonna do a little bit more math here. All right. So the target is to calculate the RAC, right? The recombination frequency between A and C. So if we treat R as a function, so we can rewrite this RAC into this R function, and the parameter and the variable for this function is XAC. What is XAC? So here I want to make it very super clear here. So the XAC represented the additive genetic distance measure, which is the number of the crossing over, right? So R is the recombination frequency function. So if you take the additive variable genetic distance in terms of the crossing over as a variable put into this function, so we can rewrite this function like this. So the R XAC equals to R parentheses. So because X is an additive measure, so we can rewrite the XAC into the sum of XAB plus the XBC, right? Okay, now this is what we have, right? What we have here, the formula we have. So we just replace this. Look at here. So we're talking about AB and BC. So we rewrite this equation using this. So this is the RAC. And because RAC equals to RXAB plus XBC, so we use the formula I just showed you in the previous slides to rewrite this formula into this way. You're gonna see C here, right? Is that clear? Okay. All right. So now let's define X equals to XAB, the distance between A and B, and we treat the distance between B and C as the increment, and we use the delta X. So this is a little bit like related to the algebra. So we define the distance between BC as the increment of the XAB, and use the delta X to represent the distance between X, B, and C as the increment. Now we can rewrite this formula into this, right? Because XAB is equals to X and XBC equals to the delta X, which is a small increment, right? Okay. Any questions, problems? The same, these two formulas are the same. I just redefine this. I define XAB, the additive distance between A and B as X. I just use X to replace this. And the distance between BC is the small, very small increment from X. I use delta X to represent this increment and replace the XBC by the delta X. And we can rewrite this formula using this. They're the same. Okay, now, so this is the formula we have. And for this equation, we just divided both sides using this delta X. And as you can see, so this is our map function, the relationship between the two the map functions here, right? And when the delta X is very small, so if you look at it here, so we can treat the RBC, we can treat the R delta X is roughly equal to the delta X itself. For example, if the region is very small, so the RBC should be equal to the XBC equals to 0. So can you follow that? So if this distance is very small, it's very small increment, delta X is very small. So we can treat these two items are equal to each other. Then we can simply use the limit theorem to do the algebra here and end up with the differential equation. This is very complicated. I will not, so in the test, in the final exam, I will not ask you to do the derivation here, but I need to make sure, make sure each of you will understand what I'm doing here. And besides, you have a open book test, right? So even you cannot memorize everything, but you can use this as a reference to help you answer the question on the final exam. So here it's important to understand this. Now using some like algebra, so we end up with this differential equation, and okay, so this is what we have, and we do the integral here, and finally we got this. So we try to find the relationship between X and Rx. So this is the map function, right? So when I show you the diagram, so that diagram is the map function, which shows the relationship between additive measure X and the non-additive measure Rx, the recombination frequency. Okay, once we have this, we can easily like rearrange this equation, put the Rx on the left and everything else on the right. And this is the map function. Okay, now look at here. So this is the general map function, but the Haldane map function is this. What's the difference between the Haldane map function and this general map function? Because we're missing a C here, right? That means in the Haldane map function, Haldane actually have a very strong, he had a very strong assumption. He assumed the C equals to 1, which means the I equals to 0, which means there's no interference at all, which means totally independent. So this works for some case, but not all the case, because in reality, for most of the situation, you're going to see some certain level of the interference between two crossing overs, right? All right. So I think for today, because this is the first lecture, I don't give you too much stuff. So I'm going to stop here. So almost 11 o'clock. So it's okay. So we have a couple of more slides. We're going to continue next Tuesday. So far, do you have any questions? Any questions about today's? Yeah, go ahead.
Can we discuss again how delta x when it's too small is the same as our function?
Sure. Okay. Here, right?
Yes.
Okay. So up to here, there's no problem, right? Okay.
Yeah. No.
Okay. So this is from the last slides. So once we this is how we describe the calculation for the recombination frequency between A and C. We end up with this formula, and then so we just simply replace XAB using X, which is the variable we want to use in the map function, right? And we use delta X to represent the distance between B and C. If we think the distance between B and C are very small, it's very small, we can treat this small distance between B and C as a extension of the distance between A and B, right? So that's why I put an increment here. So you just think about this delta X is a small increment or extension from this region. Then we can rewrite this formula using this, right?
Yep.
Okay.
Makes sense.
Okay. Yes. So because even this region is small, the delta X is small, so we can still do the math like this. So we can divide it both sides, the left hand side and the right hand side, divided both sides using this, right? That makes sense, right? Okay.
Yep.
Now the tricky part is here. So how we migrate from here to here using the Nimitt theorem. Okay. So if you look at here, so if delta X is small, so delta X is actually the same X. If you think about delta X is small, so we only look at this region, and so it almost like there's a linear regression between Rx and X. Yeah, the slope is almost like 1, but it's not that. Yeah. So let me give you a more intuitive situation. So if this region is small, just think about the region. It's very small. What is the chance for you to see a crossing over in
very
between this
small?
Yeah, very small, 0, right? And since the XBC equals to 0, and the RBC also is very close to 0, that's why we can assume they are equal. So this is always so when we deal with the differential equation, this is the common strategy we use. If you cannot look at a very large region, because this Nimitt theorem does not apply in a large region. But if you look at a very small region, the limitation, very small, you can make a lot of good and strong assumptions. So this is what we do here. Yeah. All right.
Got it.
So if it is very small, so we can assume those two things are identical, and then just do this. The limitation of this equals to this, because look at it here. So in a small region, this thing is equals to this thing. So it will become 1, right?
1.
Yes. And this will be canceled out. Then you're gonna, so for this one, if this one equals to this one, you can simply cancel out this one, and this will become this.
Oh, okay. That makes sense.
Yeah.
Thank you.
Sure.
Could you repeat that last part where you were canceling out on the limit?
Okay, sure. So this part is okay, right? So if
I get
we're talking about a very, very small region, so you're going to assume the R delta X equals to, so these two things are the same, right?
Yep.
Okay. Now look at here. So if we, from this equation to here, we just take the limitation for both sides. So this is the limitation on the left-hand side, right? And this is the limitation for each item on the right-hand side. So if you look at here, so in the limit theorem, if this delta X is very small, these two things are the same. I just mentioned here, right?
Mm-hmm.
So if you are taking the ratio of two identical things, what is the result? That's 1. Is that right?
Yep.
Okay, then look at here. So we have a couple of items here. So just ignore this part. Just look at here and here. They are identical because under the assumption, the limitation assumption, these two are identical, so we can cancel out. So, and if we cancel these two things out, you will end up with this. Right?
Got it. Okay.
Yeah, perfect. Okay. So yeah, I think it's better for me to stop here. So because I think we have covered some very solid stuff here. So you need time to digest. So I'm gonna stop here. And for today, any questions so far? Let me summarize. So it's okay. So we plan to have like another 30 minutes if you want to do the group discussion. If not, yeah, you feel free to leave and do your own stuff. So before we leave, so I'm gonna summarize. So today, so this is always what I do. So because when I summarize, so you can see the big picture of today's content, and also can help you to prepare for your final exam, right? So today, what we covered is about the maps. So we have two kind of maps: the physical map and the genetic map. This class is not very interested in the physical map. So probably other courses will give you knowledge about that. So here we are only talking about talking about genetic maps. So when we genetic map is about the measurement between two loci on the same chromosome, right? So we want to calculate the distance, genetic map distance between these two loci. There are two kind of measures. So one, we can use the average number of crossing overs between these two loci to represent how far they are removed from each other. Another measure is the recombination frequency. All right, so we have two measures. So since we can use two things to measure the same thing, so there must be some relationship between these two measures, right? So that comes the map functions, So we introduced the two famous map functions today, but we only talk about one. Next time we're gonna talk about the Kosambi. So today we only covered the Haldane map function. And this map function just shows you the nonlinear relationship between X and Rx, right? And we did a little bit algebra and the derivation to show how we start from the probability theorem, so the two regions, and calculate the probabilities of getting a recombinant progeny for the loci A and C, right? So we start from there and we do the derivation and end up with the Haldane map function. So this is what we covered so far. You're very good. Yeah, I like this class. All right, so any other questions?
I do have one question. So I am actually TAing on Tuesdays after the class. Mm-hmm. So I'm just able to join the discussions on Thursdays. Is that gonna be a problem?
No, it's no problem. So as you requested, I will record every single lecture. So if you want, if you have to do the TA and missed some part, you can watch it again.
Okay.
Yeah, don't worry.
Thank you so much.
Sure. Any other questions? Okay, so yeah, we are good to go. Oh yeah, go ahead.
Sorry, this is not about the syllabus, but do you generally give out practice exams or anything like that?
I'm sorry, could you say that again?
Sorry, do you give any practice exams or something before the finals to
Oh,
have an
yes.
idea of how
Sure,
you
sure,
know?
of course. I'm gonna give,
Okay.
yes. So this is what I usually do. So I'm gonna give you a sample test, and
Okay.
so at least you can see the styles and what
Mm-hmm.
you're gonna expect to. Of course, I will not give you the same problems, right? But
Right.
yeah, at least
Yeah.
you can see the styles and what you expect to see for your.
Okay.
Yeah.
Yeah, thank you, Dr. Jia. I come from a semester-based, university. So I'm used to taking midterms before finals. So I just wanted to know if, have an idea before the finals.
Do you want to take a midterm? I can give you a midterm if you all agree with that.
No, just the practice exam should be fine.
So I try to reduce the burden for you. So that's why I
Yeah,
don't
yeah,
give the
sure.
midterm. But yeah, definitely I will give you the practice test before the final, and it's open
Okay,
book.
yeah, that's good.
You can use a reference. So because this class, I'm gonna also give you a chance to work on some simple problems using R. So
Okay,
once you
okay.
get chance to practice on that, so I think in the final exam, I'm gonna give you one or two project. Each one has a different project.
Okay.
So to work on that,
Okay.
and you can use reference, you can use lecture notes, book textbook, so whatever. So yeah.
Okay, that helps. Thank you.
Sure. All right. So if no more question, then yeah, good to see you all. So when you have questions, just send me an email so we can schedule a discussion and a discussion between us. All right. So other than that, so nice to see you again and have a nice weekend. Bye-bye, everyone.
Thank you. Bye.
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 Mhm.
Thank you. Bye.
Bye-bye.